Picture this. Every 3 seconds, somewhere in the world, another family hears the word dementia. Money-wise, that adds up to more than $1 trillion a year. But the real cost? The memories that fade, and the goodbyes that come too soon. Here's the big question we're chasing today. Could a souped-up version of plain old vitamin K help the brain heal itself? First, we'll roll a wheelchair down a hospital corridor. Then, we'll eavesdrop on a family dinner. Finally, we'll sneak into a chemistry lab in Japan. 3 stops, 1 daring idea. Ready? Let's go! First, we'll roll a wheelchair down a hospital corridor. Then, we'll eavesdrop on a family dinner. Finally, we'll sneak into a chemistry lab in Japan. 3 stops, 1 daring idea. Ready? Let's go! Section 1. Why vitamin K? Think back to high school biology. Vitamin K shows up in the textbook chapter on blood clotting. Full stop. Yet inside your brain, tiny amounts of a special form of K quietly hang around, a few dozen nanograms per gram of tissue. When researchers tracked thousands of older adults, the folks with more of that brain-friendly K scored just a little higher on memory tests. Correlation isn't causation, but it was enough to make scientists lean forward and ask, what if? Section 2. From grocery aisle to lab bench. Here's the thing. If you want to turn a hopeful hunch into a real therapy, you can't just hand out kale and cross your fingers. You need a molecule that checks 3 boxes. 1. It has to sneak across the blood-brain barrier. 2. It has to survive the roller coaster ride of the bloodstream. And 3. It has to flip the right cellular switches without setting off alarms like runaway clotting. Enter a chemistry team in Kyoto. They grabbed a short-chain version of vitamin K called MK4. That's their Lego base. Then, they snapped on pieces that look a lot like vitamin A side chains. The result? A family of VKA hybrids. Dry names like VK2A and VK3A, sure, but each tweak, moving a double bond here, trimming a carbon tail there, changed the molecule's personality. Think designer shoes, but at the molecular scale. 1 slight stitch and the fit feels totally different. Section 3. Stem cells in a dish. Now, let's shrink ourselves down to the size of a petri dish. Scientists dripped nanomolar, so billionths of a mole, doses of these hybrids onto mouse and human neural stem cells. Under the microscope, they counted who decided to grow up and become neurons. Plain MK4 convinced about 15 out of every 100 cells. The best VKA hybrid? Roughly 45. Triple the yield, same dose, no extra cell death. Let that sink in. Section 4. Can the stuff reach a living brain? Potency on glass slides is great, but if the compound can't reach a real brain, game over. So the team injected mice with 10 milligram doses and took blood and brain samples 2 hours later. Result? Brain levels were 4 times higher than blood. Chemists call that a logP of 7.2. Translation: the molecule loves slipping into fatty brain tissue. Section 5. From chemistry to behavior. Okay, numbers are nice, but what about actual healing? In a controlled brain injury model, mice got daily injections for a week. Later, brain slices from treated animals showed thicker clusters of newborn neurons and longer axon bridges across damaged areas. Even better, when the mice ran mazes, they did a little less wall hugging and a little more correct turn-making. Not a miracle cure, just a modest uptick, but enough to raise eyebrows. Safety check. During that same week, the team watched clotting times, liver enzymes, and body weight. Nothing scary popped up, although we're talking dozens of mice, not hundreds, and 1 week, not 6 months. File that under promising, but keep watching. Section 6. Scaling the chemistry. Here's what happens next in drug discovery. Picture a 96-well plate, the kind that looks like a tiny ice cube tray. Every well holds stem cells plus a slightly tweaked VKA cousin. One variant gets an extra carbon, Another shifts a double bond. A third tweaks polarity so it hangs out 30% longer in brain tissue. Automated microscopes scan the whole plate, counting neuron markers, branch length, even synapse-like dots. If a candidate pushes neurite length from 40 to 120 micrometers and keeps 80% survival under a blast of oxidative stress, it wins a ticket to the next animal test. Do that a few rounds and you end up with a short list of hits. Molecules that might boost different flavors of brain repair, from synapse maintenance to myelin rebuilding. Section 7. The Human Hurdle. You can probably guess the next step: early human trials. Picture 80 adults with mild cognitive impairment, half on the hybrid, half on placebo, all watched over 24 months. Regulators will want hard data. Did hippocampal volume shrink more slowly? Did yearly memory decline drop by at least a quarter? In stroke or spinal cord injury, even a 15% bump in motor score can mean the difference between needing a wheelchair and walking with a cane. Section 8. What you can do right now. While the labs grind away, there's low-hanging fruit you control today. Manage that blood pressure. Aim for 150 minutes of movement a week. Grab 7 to 9 hours of good sleep. Each habit alone shaves dementia risk by 10 to 20%. Stack them, and you're already way ahead. If VKA-style drugs someday add another 15% delay, that's the difference between spending years in a memory care unit and years enjoying grandkids' soccer games. Mini challenge. Want a fun experiment? For the next 7 days, add 1 vitamin K-rich food, steamed kale, spinach, broccoli, or for the adventurous, fermented natto, to a meal you already eat. Right after dinner, do a 10-minute online working memory game. Keep your coffee, sleep, and exercise routines steady so you're only changing the vitamin K piece. At week's end, compare your scores and jot down how mentally sharp you felt. No, it's not a clinical trial, but it is a peek at how small tweaks may play out in your own brain. Take a breath. We've covered a lot. Final recap. So here's what we unpacked today. 1. Vitamin K isn't just for clotting. Tiny amounts already live in your brain, and higher levels link to better memory. 2. Japanese chemists built VKA hybrids by fusing vitamin K and A features, creating molecules that slip into the brain and talk to neural stem cells. 3. In dishes and in live mice, the best hybrid tripled neuron formation and nudged injured brains toward repair, with no major safety red flags in the short term. 4. A high-speed mini-factory approach could spin out even better versions, each targeting a different piece of brain resilience. 5. While human trials gear up, everyday habits, exercise, sleep, blood pressure control, and maybe a few extra leafy greens, are powerful tools you already own. Keep an eye on this space, because the next breakthrough might start with something as ordinary as a vitamin. Thanks for listening, and as always, stay curious and stay kind to your future brain.